Okay. Thank you all. Thank you all for coming. It's an annual enormous pleasure to introduce the Gil Blount Memorial Lecture and the Gil Blount Memorial Lecturer. Let me begin by saying a few words about Gil and then a few words about Dr. Wu, who is our speaker today. Gil Blount, whose picture is up there, was the inaugural chair of and founder of the Division of Cardiology. And he served as division head for an astounding 30 plus years, which is a testament to his stamina, commitment, and I don't know what else. His accomplishments were extraordinary. When he first moved from Hopkins to Colorado, he founded our cardiac catheterization lab. He founded the CVP, which is currently directed and has survived all these years by the director is sitting in, the current director is sitting in the back of the room. So that institute has continued and prospered. Gil was very interested in congenital heart disease and in the effects of hypoxia on the heart. In Baltimore, he had trained with, amongst others, Helen Tausig and Richard Bing. So he brought an appreciation of congenital heart disease and of cardiac physiology with him when he moved across country. In his 30 years, his accomplishments, as I mentioned, were exceptional. And he himself reported that the thing that he was most proud of was the fact that he had trained probably 2 generations of cardiologists who populated the state of Colorado. And it's annually an enormous pleasure to acknowledge some of Gil's trainees who are sitting in the 1, 2, 3, 4th row, who are co-sponsors of this lecture. So thank you, gentlemen, for coming. In addition to his trainees, Gil was extremely fond of his family of 5 girls, itself something of an accomplishment. And we are also annually blessed by Gil's family, and Donna is sitting in the back of the room. So we also are delighted that that she comes to this lecture. So it really is an acknowledgement of an enormous legacy to the division, the department, and to the state of Colorado. And we are exceptionally proud annually to host this lectureship. Our lecturer this year is Joe Wu. Joe Wu is really an exceptional cardiologist. He currently is the Simon Stertzer Chair and Director of the Cardiovascular Institute at Stanford. His accomplishments are frankly dazzling. In his relatively short career, he's published over 600 papers, and I think he's one of the top 10 cited cardiologists in the literature. He's received basically every award that the profession can offer, from an NIH Clinical Pilot Award, Early Career Award from the American Heart Association. He's a member of the ASCI, the AAP, the American Association for the Advancement of Science, and the National Academy of Medicine. He's also, for what it's worth, and I don't know if this is an honor or a curse, he's the current president of the American Heart Association. His work has been exceptional, exceptionally innovative. He's used iPS cells as a platform to model and study complex cardiac disease, including congenital heart disease. And the work, it really has been, frankly, inspirational. In addition to being a visionary scientist, Joe's also an extremely nice guy, very generous with his expertise, and he himself has launched the careers of probably 50 or 60 junior cardiologists, several of whom are on our faculty. So we thank him, in addition for his scientific accomplishments, for his contribution to our division and department. It really is an extraordinary pleasure to acknowledge Dr. Blount and to introduce Dr. Wu.
I think I'll just use the mic here. Yeah. Can you hear me okay? Yes. So I want to thank Peter for the very nice, wonderful introduction. I also want to thank Larry for inviting me here, and also Dr. Chopra, for inviting me to this special lecture. And I also want to again thank the daughter of Dr. Blount for being here. And as Peter mentioned, Dr. Blount is being remembered by his family, friends, and colleagues for his many contributions at the cardiovascular field. And I was looking this up last night, including congenital heart disease, pulmonary hypertension, high altitude physiology, and others. And I was actually trained as an adult cardiologist who sees patients in adult congenital heart disease. When I first showed up at Stanford, those were the patients I was seeing. So here are my disclosure slides for my talk. And I want to start the talk by giving the audience a brief clinical case report that I saw at Stanford and how it ties together this concept of stem cells, genomics, and clinical trial in a dish. And so this is a 57-year-old man, previously healthy, came to our clinic for newly diagnosed non-ischemic cardiomyopathy, found to have severely reduced ejection fraction. And what's interesting is that the family is remarkable for multiple sudden cardiac deaths, atrial fibrillation, heart failure, hypertension. And because of the family history, we decided to do a DNA sequencing, and it shows a pathogenic variant of lamin AC mutation. So the question is, what is the mechanism of this disease? How does this genetic information affect his management right now, quote unquote, precision medicine? And how do we treat it outside of standard care and also in the future? So just keep this case in mind. And I think as many of you know, there's a significant interest in precision medicine. I think this concept was first started by President Obama back in 2015. And much of this is made possible by recent advances in next generation sequencing, bioinformatics, and big data. The concept is that by understanding our genes, along with our lifestyle environment, we should be able to better predict which treatments might work best for us. Now, in my opinion, I think there are three key technologies that marries precision medicine and basic science research. The first technology is DNA sequencing. I think, as you know, this technology has become much cheaper with lower error rate, broader coverage, much faster turnaround time. For example, back in 2000, it would cost you $1 billion in over 13 years to get your DNA sequence. Today, you could get your DNA sequence for less than $1,000, essentially about $400 right now, and within 3 to 4 weeks turnaround time. And then, in my opinion, I think the second key technological breakthrough is the various types of genome editing technologies, starting first with zinc finger nucleases, and then TALEN, and then culminating with the CRISPR-Cas9. This was discovered by Emmanuel Charpentier and Jennifer Doudna back in 2011. And so using these genetic scissors, you can now change the DNA of animals, plants, microorganisms, and stem cells with extremely high precision that allows you to validate genetic mutations and variants. And just this past December, for example, the FDA had approved its first CRISPR-based gene therapy for treatment of patients with sickle cell disease. I was actually on the FDA Cell Tissue and Gene Therapy Advisory Panel that reviewed the data that was presented to us by Vertex Pharmaceuticals. And in my opinion, I think the third key technological breakthrough is our understanding of human stem cell biology. And for the non-stem cell biologists in the room, I will broadly categorize this into 3 cell types. The first type of stem cells is adult stem cells. These are found in tissues in adults, children, and also neonates. These cells are pretty much committed to become a cell from their tissue of origin. For example, stem cells in the bone marrow can give rise to red blood cell, white blood cell, but they cannot give rise to brain cells or heart cells. Likewise, the stem cells in the brain can give rise to neurons, but they cannot give rise to hematopoietic stem cells. Here, the key thing to keep in mind is that as you get older, your adult stem cells get depleted. This results in a multitude of medical problems, such as dementia, sarcopenia, osteoporosis, skin wrinkles, and white hair, which I have a lot these days as I'm getting old. The second cell type in our body is actually not in our body, but the second cell type is human embryonic stem cells. These cells were first isolated by Jamie Thompson's group back in 1998. This only exists at the very earliest stage of embryonic development, usually around day 4, day 5. And then unlike these adult stem cells, ES cells can essentially become pretty much all the cell types in your body, and that's why they're called pluripotent. And unlike ES cells, unlike adult stem cells, these ES cells also continue to divide. That's why they have this self-renewal property. But as you know, work with ES cells is hampered by ethical and political controversies. And then the third key cell type that I want to focus most of my talk on is this concept of induced pluripotent stem cells. This was first discovered by Shinya Yamanaka around 2006. Because these iPS cells, we essentially take your blood or take your skin, hit it with 4 reprogramming factors, make these iPS cells that are for all practical purposes the same as your embryonic stem cells. So once we generate with your embryonic stem cells, but we call it iPS cells, you can then take these iPS cells and differentiate into all cell types in the body. And here, for example, I just highlighted that we've developed protocols to generate iPS cell-derived heart cells, iPS cell-derived cardiac fibroblasts, smooth muscle cells, endothelial cells, and cardiac pericytes, the big 5 cell types in the human heart. Once you generate these different cell types, you should be able to perform disease modeling. You should be able to do drug discovery research, and you should be able to do what we call a clinical trial in a dish model. So over the past 10 years, my lab at Stanford, in collaboration with Greenstone Biosciences, we made iPS cells for more than 2,000 patients with different medical history, ethnicity, and sex. These cells are linked to medical information. We perform multi-omic sequencing to help us understand how human genetic variants impact the drug response phenotypes. We also work with the FDA on drug safety testing, and we share these cells freely with scientific community. For example, we've had a very fruitful collaborations with Drs. Luisa Mastroni, who I just saw, Matthew Taylor, and others here at Anschutz. And then today, we've also sent out more than 4,000 vials of these cells to more than 400 investigators nationwide. And so this is our typical workflow. We recruit patients from our clinic at Stanford, as well as outside of Stanford. We then generate their 2D micro pattern human cardiomyocytes. We can also generate the 3D human cardio organoids and 3D human engineered heart tissues. And because these are derived from the patients, genetically, they're essentially the same as the patients that we generated the iPS cells from. And now, besides cardiomyocytes, as I mentioned in the earlier earlier slides, these iPS cells can be used to differentiate to other cell types. For example, we can differentiate them to mesoderm by GSK inhibition, into cardiovascular progenitor cells by Wnt inhibition, into cardiomyocytes by Wnt activation, into cardiac fibroblasts by TGF-beta inhibition, into smooth muscle cells by PDGF activation, and into endothelial cells, as shown here, by VEGF addition. Now, all these have been worked out in the past 20 years. For example, back in 2004, when I first started, we were only getting about 1 to 2% efficiency of cardiomyocyte differentiation. Today, we can consistently get 90% plus cardiomyocyte differentiation without any issues. And as I mentioned, we've also worked out the protocols for endothelial cells, smooth muscle cells, cardiac pericytes, and cardiac fibroblasts as well. And so with these 3 sets of tools, the genetics, the genome editing, and also the stem cell biology, you should be able to study cardiovascular genetics and epigenetics in detail. And this is the main focus of my lab. For example, we now know that there are many different types of genetic changes, such as single nucleotide polymorphism, copy number variation, and also chromosomal aberrations. We also know that there are many different types of epigenetic changes, such as histone modification, DNA methylation, and also non-coding RNA. These genetic and epigenetic changes then play important roles in causing coronary artery disease, cardiac arrhythmias, cardiomyopathy, aortopathy, dyslipidemia, and pharmacogenomics. So now I'll give you several examples very quickly of how we combine genetics, genome editing, and iPSCs to study these diseases. One first example is a familial dilated cardiomyopathy. This is frequently associated with undiagnosed or idiopathic heart failure. I think a lot of seminal work were actually done here by Mike Bristow in the audience here. And so this is one of the earliest examples of how we use iPSCs to model this familial DCM, and to also use iPSC cardiomyocytes as an in vitro platform for drug screening. In this study here, we generated iPSCs from a 3-generation family with DCM due to a point mutation in cardiac troponin T with the arginine to tryptophan switch, codon 173. This kid here already had a heart transplant at age 15. And so when we compare the healthy versus the disease cardiomyocytes that we generated from this family, we were able to show that the DCM cardiomyocytes had altered regulation of the calcium ion, decreased contractility, and also abnormal distribution of the sarcomere alpha actinin. And importantly, when we treated with the drugs such as beta blocker, again, a lot of similar work was done here. These beta blockers can improve the function of the DCM iPSC cardiomyocytes, recapitulating results from multiple large beta blocker clinical trials. This is a follow-up study. Here, we will refer a large family to our clinic with familial hypertrophic cardiomyopathy, or HCM. This occurs in about 1 in 500 patients. In this case here, it's due to a myosin heavy chain 7 mutation with the arginine to histidine switch at codon 663. The mother here had manifested HCM phenotype with the cardiac arrhythmias and hypertrophy and the cardiac MRI. She has 8 kids. The 2 oldest are already manifested the HCM phenotype, whereas kid number 3 and kid number 8, they were genotype positive, but phenotype negative, most likely because they were still young at that time. And so through a whole series of experiments, we were able to understand mechanistically how this myosin heavy chain 7 mutation causes increased intracellular calcium, which leads to calcium handling defects and then cellular arrhythmia. It also causes activation of the calcineurin and fat signaling pathway, which leads to hypertrophic responses. And again, we use this as an in vitro setup for drug screening, showing that treatment with a calcium channel blocker, such as verapamil, or a beta blocker, such as propafenone, can be used to reverse both the cellular arrhythmia as well as the cellular hypertrophy. Now, in pediatric population, the 3rd most common type of cardiomyopathy is left ventricular non-compaction, which occurs in about 1 in 1,000 patients. And so this is a follow-up study that we will refer this large family at Stanford, and also a smaller family from Japan. The boy here at Stanford already had a heart transplant. His older brother and older sister also had LV non-compaction, but were asymptomatic. And his father had mild dilated cardiomyopathy without LVNC, and his paternal grandmother also had the cardiac arrhythmia requiring a pacemaker. And so through a whole series of experiments, we're able to confirm that the mutations in cardiac transcription factor, TBX20, as shown here, one in the T-box domain for the Stanford boy, and in the other in the transactivating domain for the Japanese girl, causes abnormal activation of the TGF beta signaling pathway, which recapitulates a key aspect of the LVNC physiology in this family. Now, previously, I have shown you that the iPSCs can be used to model Mendelian mutations, such as troponin T in DCM, myosin heavy chain 7 in HCM, T-box 20 mutation in LV non-compaction. In addition, these iPSCs can also be used to study the impact of single nucleotide polymorphism. For example, some of you may or may not know that the most common Asian flush syndrome is actually due to a genetic variant in the aldehyde dehydrogenase. It is one of the most common genetic polymorphism in the world. It counts about 36% among East Asians and about 8% of the world population. And again, it causes this alcohol flush reaction, which includes flushing, nausea, headache, and tachycardia. We actually treat alcoholics by giving them antabuse or disulfiram, which works on similar mechanism and is used to help alcoholics stop drinking. Now, interestingly, this aldehyde dehydrogenase polymorphism involving glutamic acid to lysine switch, this has been linked to increased risk of coronary artery disease, more severe outcomes after heart attack, higher incidence of hypertension and cancer, and also increased complications from type 2 diabetes. So I actually don't have this particular enzyme. And so if I drink a half a glass of wine, my face will look like this in about 10, 15 minutes. It's actually a good enzyme to be missing because it's hard for me to be alcoholic. And it's also a good enzyme to be missing because when I'm traveling, I'm not in the gym. I don't need to go to the gym. I just need to drink half a glass and my heart rate will shoot up from 60 to 120, stayed up for about 30 minutes and then comes down. Now, joking aside, this enzyme deficiency has actually been well documented to be a risk factor for coronary artery disease and myocardial infarction in East Asians. For example, here you see in Japanese men, Korean men, and also in Han Chinese. So when I saw these epidemiology data, I had to prioritize it. And I asked my postdocs to work on a stat, right? And so this is a study we published about 10 years ago. So what we did was we recruited 10 Stanford undergrads who are of East Asian descent. 5 of them are wild type, which means they can drink no problem. 5 of them are heterozygous, which means they have similar symptoms like I do. And if you're homozygous, missing both copies, which is about 1% among East Asians, you can not really touch alcohol. You have a very severe reaction. And so here we modeled the myocardial infarction by exposing the cardiomyocytes to hypoxia. And as you can see here, the wild type cardiomyocytes tolerated the hypoxia quite well with minimal apoptosis, whereas the heterozygous cardiomyocytes has much more apoptosis with the same amount of hypoxia. So to make a very long story short, overall, we believe that the enzyme is involved not only in detoxifying alcohol, but also getting rid of a reactive oxygen species, or ROS. After a heart attack, there'll be tons of ROS that gets activated. If you're missing this enzyme, you have a harder time getting rid of ROS, and hence leading to more cell death, and perhaps explaining for the worst clinical outcome that are observed in epidemiology studies. Now, we also know from large genome-wide association or GWAS studies using the Biobank Japan data set that this enzyme deficiency is one of the strongest SNP associated with coronary artery disease. So in a more recent follow-up study, we used the iPSL-derived endothelial cells, not the cardiomyocytes, but the endothelial cells from these ALDH deficient patients, exposed them to alcohol to model the vascular dysfunction and the CAD. And importantly, we also did a compound library screen and also a molecular docking analysis to show that the SGLT2 inhibitor, empagliflozin, can actually mitigate the vascular dysfunction. And empagliflozin does this by alleviating the ALDH-induced oxidative stress and nitric oxide reduction by inhibiting the sodium hydrogen antiporter 1 and also activating the AKT-ENOS pathway. And then these findings were also confirmed in mouse models of diabetes and alcohol exposure. We didn't pursue this as a target, mainly because I didn't want to encourage people to drink more wine and alcohol by saying, oh, I can take EMPA to mitigate the vascular dysfunction. But this is just a proof of principle that you can use this concept to screen for drugs that can mitigate the vascular dysfunction with alcohol. So these are 2 examples. We tested it in the East Asians. But you should be able to use this platform to study other genetic susceptibility among different ethnicities. For example, from a health and science perspective, we're interested in understanding why African Americans have much more hypertension, why Hispanics have much more diabetic vasculopathy, and why South Asians, for example, Indians and Pakistanians, have much more metabolic syndrome. So I want to switch gear now to the second part of my talk, which is using this iPSL platform, genetics and genome editing for drug screening. As you know, this process is actually quite time-consuming and expensive. For example, typically it'll cost you about $1 to $2 billion in 10 to 15 years to bring a new drug from the discovery phase to the clinical trials to market approval. Now, what some of you may not know is that many of the drugs that you take today actually do not work at all. So shown here the top 10 highest sales of drugs in the US. Blue means the drug work and red means the drug does not work. So for Abilify, which is used to treat schizophrenia, out of 5 people, the drug only helps 1 person. For Nexium, which is used to treat heartburn, out of 25 people, the drug only helps 1 person. So overall, this is pretty poor response rate, and this is the reason why the editorial here is titled Time for 1 Person Clinical Trial. This is not surprising because essentially this is equivalent of number needed to treat, NNT. So for cardiology, for example, for the drugs that we give for heart failure, ACE inhibitors, spironolactone, and SGLT2 inhibitors and so forth, the NNT is typically around 30 and 40 range. Now, in the US, both the FDA and the big pharma are also thinking about using these iPS cells to accelerate the drug development process. This is a study published by several companies and the FDA in which they use these iPS cell-derived cardiomyocyte to predict arrhythmia risk as part of the comprehensive in vitro proarrhythmia assay initiative. I think some of you may know all the drugs that we take today, they need to be tested for cardiac arrhythmia before they can get approved by the FDA. Currently, the platform is testing the drugs on CHO cells or Chinese hamster ovary cells or HEX cells, human embryonic kidney cells, overexpressed in the hERG channel. Not a very ideal scenario because the CHO cells and HEX cells are not cardiomyocytes. And here the investigators are testing drugs on using cardiomyocytes instead. They use 1 consensus protocol, 2 human cell lines, 3 continents, 28 blinded drugs, 2 statistical models and 10 volunteer labs. Importantly, they are able to show an 87% prediction rate, which is very good considering that this is one of the earliest studies by the FDA, and therefore there's going to be significant room for improvement in the future. Now, in our lab, we've also been pushing that direction as well. This is a paper we published in 2016 using transcriptomic profiling of iPSC cardiomyocyte to predict individuals' drug safety and efficacy responses in vitro. Here, we tested 2 drugs, rosiglitazone and tacrolimus. We also use CRISPR genome editing to help us confirm the potential mechanism of action as well as the adverse effects, especially for rosiglitazone. This is a PPAR gamma agonist. This was used to treat diabetic patients in around 2000, when I was a health staff at that time, but was pulled off the market in 2020 because of increased cardiovascular risks. And so one of the goals in my lab is to start creating what we call a human encyclopedia. A human encyclopedia of drug response signatures using this model. And so showing here 2 examples whereby we assess the effects of 4 different kinds of calcium channel blockers, nifedipine, amlodipine, diltiazem, verapamil, 4 different kinds of statins, atorvastatin, lovastatin, simvastatin, fluvastatin. And what is interesting is that you will see different gene expression patterns among these drugs. And this suggests that even though they belong to the same class of drugs, they may exert very, very different effects among different individuals when all of us take these medications. And so this is a statin story. And so in a more recent follow-up on the statin story, we actually use the iPS cells and multi-omics to dig deeper into the role of how statins protect vascular health. Now, as you know, statins are given to over 40 million adults in the US alone, including myself. I'm taking atorvastatin, which is a Lipitor. My primary care doctor just checks my LDL. But I ask her, what is the effect of statin on my brain, on my heart, on my vascular? Nobody knows, right? So there's a lot we still don't know about how these drugs work. And so here, we show that the statin can improve endothelial function by suppressing epigenetic-driven endothelial to mesenchymal transition. And what we did was we took iPS cells, generated endothelial cells, exposed them to simvastatin. We used ATAC-seq and observed changes in the gene accessibility in these iPS cell endothelial cells. And we found that the simvastatin can protect the endothelial cells from endothelial dysfunction by epigenetic regulation of the YAP pathway. And the YAP pathway is involved in genes that are in the endothelial to mesenchymal transition process. And again, we validated this in a diabetic mouse model and also confirmed it in an in vivo setting. Now, as you all know, besides cardiology, there's also significant advancements in chemotherapy drugs, and that's because cancer and heart are the top two causes of morbidity and mortality in the US. As more cancer patients survive due to better immunotherapy and chemotherapy, more patients end up with heart failure symptoms due to cardiac toxicity from these drugs. And this is evident by the dramatic increase in terms of the cardio-oncology clinics in major academic centers, including Stanford. We recently set up a cardio-oncology clinic, and we now have 3 cardio-oncologists right now. And so we've been working on this for the past 10 years, and showing here's a paper we published about 8 years ago in which we use iPS cells to predict chemotherapy-induced cardiac toxicity. And here we recruited 8 breast cancer patients. A year later, some of them had normal cardiac function while others had doxorubicin-induced cardiac toxicity. We then generated their cardiomyocytes, exposed them to increase in dose of doxorubicin, and we then work out the mechanism by studying their contractility, calcium cycling, metabolism, gene expression, apoptosis, and different assays. And here, as a proof of principle, we show that if we pick the patients appropriately, these iPS cells can be used to recapitulate which patients are much more susceptible to doxorubicin-induced cardiac toxicity. Right now, we just don't have a good guess, right? It typically occurs in about 8 to 10% of our patients who get doxorubicin. Now, this is a follow-up study in which instead of doxorubicin, we focus on another popular chemotherapy drugs, which is a tyrosine kinase inhibitors. And these tyrosine kinase inhibitors, they kill cancer cells by inhibiting the phosphorylation activity of hyperactive receptor tyrosine kinases. And common TKIs you may have heard of include Gleevec and Sutent, which inhibit VEGFR2 and PDGFRB in cancer cells, but they can also cause heart failure as a side effect as shown here. And this is because both VEGF and PDGF signaling pathways are critical regulators of cardiovascular development. And so in this study, we generated iPS cells from 13 patients, treated them with 21 most common TKIs, came up with a cardiac safety index that allows us to predict which TKI is cardiotoxic and which TKI is not. So our ultimate goal is to further optimize this cardiac safety index so that we can better predict the safety profiles of chemotherapy drugs before giving them to patients. And so as you can see from these examples I was just giving you, we are integrating iPS cell biology with various cellular, molecular, and functional readouts, as well as multi-omics to help us better understand the disease at the systems biology level. I think in the next few years, you'll see more examples of investigators using AI machine learning, large-scale iPS cell populations to study different cell types, and also to study expressing quantitative trait loci, or EQTL. And now these days, you cannot give a talk without saying something about AI machine learning because of its broad impact in biomedical science. For example, you can now use AI to help you interpret EKG patterns, to assess LVEF and echo and MRI, and also to monitor cardiac arrhythmias using wearable devices. At the same time, in the drug discovery space, there's also significant advancements using AI. For example, working in collaboration with computational scientists at Greenstone Biosciences, we routinely use AI machine learning softwares that help us analyze the protein structure by in silico molecular docking. Once we know the molecular target and the protein structure we're going after, we can then do virtual screening of, say, about 1 billion compounds at a time. This will take us about 2 months to screen for all these 1 billion compounds. So this is a schematic of our typical molecular docking campaign. You can start with the virtual compound, or you can start with existing compound, such as the Zinc-22 library, the FDA repurposed library, the enamine library, and also the WuXi chemical library. Alternatively, you can also perform a smaller sampling size, to say 100,000 compounds at a time for your molecular docking campaign. Once you identify the molecular docking site, you can further refine the rank score, as well as perform autodock and molecular dynamic simulation to look for compounds with desired drug-like properties that you're going after. So here, I just want to highlight one recent example using AI machine learning and in silico docking for drug discovery. I think, as you all know, marijuana is being used increasingly in the US due to legalization. I believe the two states with the highest use of marijuana is probably Colorado and California. Interestingly, when we look at the UK Biobank database, we actually found that the marijuana use is actually associated with increased myocardial infarction. However, the exact mechanism is unknown. And here we show that the delta-9 THC, this is a psychedelic component of marijuana, can actually bind to the cannabinoid 1 receptor, or CB1, and then cause vascular inflammation, oxidative stress by activating the MAP kinase and the kappa B pathways, and also a whole bunch of cytokine pathways. This then leads to oxidative stress, inflammation, and cell death. And then importantly, using AI machine learning, as well as in silico chemical library screening, for example, the slides that I show you, we found that ginestin serves as a neutral CB1 antagonist that can competitively block the binding of THC to the CB1 receptor, and hence block the harmful cardiovascular effects of THC, as shown in this cartoon. Ginestin is actually a flavonoid. It's found abundantly in soybean. So my former postdoc, Mark Chandy, who's now a faculty in Canada, is now doing a clinical trial by giving ginestin to chronic marijuana users. So for the last part of my talk, I just want to then touch upon how we can then use these iPS cells to implement the concept of clinical trial in a dish. Here, the concept is that you should be able to recruit patients, generate their differentiated cell types of interest, test which patients respond to drugs and which patients do not by using the iPS cells as a surrogate, and then go back to perform the actual trial on patients who are positive responders to the drug. And that way you have a higher chance for your phase 1-2 clinical studies, decrease the cost, and also accelerate the timeline for drug development. This concept is actually quite timely, because some of you may know the FDA Modernization Act. This was passed by both the Senate and the House on December 2022. Think about when was the last time the Senate and the House agree on something unanimously, right? Very rare. And so this law authorizes the FDA to use alternative testing platforms, such as stem cells, organoids, and in silico modeling, and for some drugs to completely eliminate the requirement of animal testing before going into clinical studies. The goal here is to improve the predictiveness of preclinical safety and efficacy assessment, and also to reduce the number of animals in drug discovery and development program. This slide highlights, in my opinion, what I think is one of the major problems with the high failure rate in drug discovery, and that is the lack of genetic diversity and pharmacogenomic differences with traditional models, such as using immortalized cell line and also using genetic inbred mouse. For example, if you stop and think about it, because there's immortalized in inbred, the differences that you see here between your control and treatment group, it's actually due to the technician's pipetting skill for cell culture work, or due to the technician's tail vein injection skill for animal work. On the other hand, the differences that you see here in a population of, say, 1,000 diverse iPS cell lines is actually due to the underlying genetic variability that exists among all of us here. And ultimately, this pharmacogenomic difference is what you're trying to understand before you go into clinical studies that is otherwise impossible to capture or recapitulate using one cell line or one inbred mouse models. And so, as I mentioned earlier, I'm on the FDA Cell Tissue and Gene Therapy Advisory Panel, and this slide is given to me by my colleagues from the FDA for rare orphan diseases in which it's very hard to recruit patients. The FDA recognizes the challenges of developing new drugs due to small number of patients, and yet there are thousands of genetic variants for each disease. So now their approach is to start accepting data generated from cell models to test drug efficacy. So as an example, for cystic fibrosis, the drug Callidico, made by Vertex Pharmaceuticals, initially approved for 10 genetic variants, and now has been expanded to 24 more genetic variants. Another drug is Fabry disease. This is a lysosomal storage disease that is passed on by the X chromosome. The drug Galafold, made by Amicus Therapeutics, initially approved for 40 genetic variants and now has been expanded to 348 genetic variants based on cell models. And so currently the FDA is interested in expanding the cell-based platforms across the full spectrum of diseases. And so this slide is also given to me by the FDA. I think the value proposition here is using these in vitro efficacy data to help us reduce the number of clinical studies, to inform the regulatory decision-making at the time of marketing application, to streamline the drug development process, and also to expand the indications based on these data. So with that, I want to go back to the case I mentioned about this gentleman who was diagnosed to have non-ischemic cardiomyopathy. DNA sequencing shows Lammin mutation. And the question is, what is the mechanism of how Lammin mutation causes this cardiomyopathy, and why is it there's so many people with these issues? So it turns out this patient belongs to a very large family cohort with Lammin AC frameshift mutation that leads to early termination of Lammin, and the Lammin is essentially a nuclear envelope protein. Lammin cardiomyopathy accounts for about 10% of familial DCM. It is the second most common cause of genetic cardiomyopathy, dilated cardiomyopathy, with Titin mutation being the most common type. As you can see here in this large family, many of the affected individuals had cardiomyopathy, atrial fibrillation, atrial ventricular block. Importantly, eight of them died from sudden cardiac death. And so this is a terrible, exerts a terrible toll on this family. This was referred to us. And here we figure out that the mechanism is actually due to abnormal activation of the PDGF in the cardiomyocytes, which then leads to cellular arrhythmia and also impaired contractility, as shown in this cartoon on the right. We also show that blocking the PDGF pathway with the PDGF inhibitor, such as quinolinib, a very low dosage can rescue the abnormal phenotype of these cardiomyocytes. Now, we actually look into quinolinib. The problem is quinolinib is not yet an FDA-approved drug. It is owned by Pfizer, which licensed it to AROC, and they're at phase 3 trial for AML. And so we're not able to pursue this further. But what's interesting is that at the same time, we also noticed that many of our Lammin disease patients, they had early onset hypertension at age 30s and 40s. We were quite puzzled by this because typically you would expect somebody with low EF to have low blood pressure, not to have a high blood pressure. So we suspect the mutation may have something to do on the endothelial cells or vascular cells. And here we decided to differentiate the iPS cells into vascular endothelial cells, compare the differences between the healthy and disease. To make a long story short, we identified KRF2 as a transcription factor that is responsible for the endothelial dysfunction. We then screened for FDA-approved library. The previous one, we screened for just a small compound library. And we found that lovastatin can be used to activate the KRF2 and rescue the endothelial dysfunction. And as I mentioned, in contrast to quinolinib, lovastatin is FDA-approved, and we can repurpose it for these patients. And that's why here the title is Clinical Trial in a Dish using iPSL shows that lovastatin can improve endothelial dysfunction and cellular crosstalks. And so the same Lammin patients whom we tested their endothelial cells were asked to return to our clinic, were given lovastatin daily, even though their cholesterol numbers are completely normal. The clinical data shown here, we use the EndoPAT, which is a machine that assesses flow-mediated dilatation during reactive hyperemia. Normal index is 1.7 and above. And basically you include the brachial and you release it to see how much blood flow come back. And as you can see here, this Lammin patients, number 2 started with a very poor index at 0.48, improved significantly to 1.06 after 6 months of lovastatin treatment. So I think this is a clear example of what we call a clinical trial in a dish, whereby we first identify a large family Lammin mutation, elucidated the mechanism of vascular dysfunction. We then screened for a panel of FDA-approved drugs using this patient-specific cells, found that lovastatin can improve the cellular phenotype by enhancing the KRF2 pathway. We then confirmed the in vitro findings in the actual patients themselves by using EndoPAT to show functional improvement. So I believe the FDA Modernization Act will really pave the way for the next era of drug discovery using predictive AI, generative AI to find the optimal candidate, similar to the story I showed you earlier, involving genestin as a neutral CB1 antagonist to mitigate marijuana-induced vascular toxicity. As you can see here, whether these are drugs, peptides, antisense oligonucleotides, or RNA therapeutics, you can then test them in vitro, as well as test them in vivo with the traditional PK/PD studies in mice, rabbits, or large animals. However, before going to the patient themselves, you can actually perform what we call clinical trial in a dish model that will help you better predict the safety, efficacy profile of your lead compounds prior to going to these clinical studies. So this is my last slide. I hope I have shown you the framework of how there may be a paradigm shift in terms of how we use patient-specific iPSLs to help us better understand, first, the mechanisms of action. This is biological data right here. These biological data can then be converted into digital data with multi-omics sequencing. These digital data can then be fed into AI machine learning software for your molecular docking, molecular simulations, and also in silico ADMET prediction. This will then give us drugs that are much more precise and predictive. The lead compound can then be tested on the patient-specific iPSL derivatives that have the desired phenotypes that you designed the drug for, and hence completing this loop of clinical trial in a dish. The ultimate goal is to help decrease the cost of drug development, shorten the timeline, and develop therapeutics that are much more efficacious and safe for our patients, which is the holy grail for precision medicine. So this concludes my talk. I want to acknowledge my postdocs, graduate students, instructors, my collaborators, and my former trainees. I think we've had several come by here, including Edward Lau, and also my collaborators at Stanford, outside of Stanford. Special shout out to Louisa, Matthew, Sunit, whom we collaborate closely with, as well as our funding support from the NIH, AHA, Chen Zuckerberg, FDA, and my collaboration with Greenstone. Thank you so much again for this invitation. Thank you.
No, that was a thrilling talk. Thank you so much. I think we have time for a few questions. I think traditionally we should ask trainees if they have questions first and then elevate to more senior people. You're going to have to shout.
Might need to repeat what about Mike. Yeah.
I think he was asking the question about the dangers of scientific discovery and how we can develop race-based therapeutic strategies which might actually be harmful rather than beneficial. Is that?
I can try with a microphone.
Yeah, you try.
So we obviously have a pretty ugly history as a country around like biological essentialism and, race-based guidelines that do harm. And so my question is, what steps do you feel like we need to take as a scientific community to make sure that this sort of work doesn't contribute to that, but that we're still able to harness its immense potential?
So I'm gonna have a hard time.
The question relates to basically the dangers of scientific discovery, and can these be used in ways that are harmful rather than beneficial?
Oh, the dangers.
How did I do?
Yeah, so maybe we could talk offline. I think we always want to. I think if you're referring to the dangers of scientific discovery, you're probably, I don't know which areas of dangers of scientific discovery. Maybe the DNA sequencing, that's one aspect. The genome editing is another aspect in terms of quote unquote designer babies. I think for the other aspect using These are iPS cells to help us predict what's going to happen to the patients. I think, I talked to Petit here, and she's a bioethics. And so we as physicians, we just need to work closely with our colleagues and also with our patients and also with our bioethicists to see what's feasible and what's not feasible. I'll give you the example. People have done genome editing, the one that's FDA approved. Sickle cell, patients with sickle cell, you take out the hematopoietic cells, genome edit, and infuse the cells back to them, helps the patients a lot. That's fine, but I think maybe about 4 years ago, there was a group in which they took the embryo and genome edit the receptor that the HIV virus targets, and then create quote unquote babies that no longer have the receptor. Is that fine? Probably not fine 5 years ago. But is it fine 10, 20 years from now? We don't know in terms of these are designer babies. So there's a lot of discussion. But at the same time, I think this is really what's fascinating about scientific discoveries, because you push the boundaries of what's okay and what's not okay. 60s, I believe it's about 60 years ago when IVF came about, there was a lot of discussion about IVF as well, in vitro fertilization. These days, look at how much IVF has helped couples who have trouble getting pregnant. Yeah.
Other questions? Yes.
Maybe you can speak up louder. Yeah, that's good.
We got a microphone coming your way.
I'm just curious your thoughts. Should all of us be paying $400 to get our genome sequenced?
Yeah, $400. Yeah. I got I'm not sure how many of you got the DNA sequence. I've got my DNA sequence.
Right.
Yeah.
So what are your thoughts on like the average person going out and that being part of something that you do for your own healthcare, going out and getting that sequenced?
What was the question?
Did everybody get their DNA sequenced?
Oh, yeah. So that's a tough question, right? And I do believe right now there's a lot of pros and cons about getting your DNA sequence, whether, what happens if you have something and then it's on your medical record and so forth. I do think that in the future, 20 years from now, maybe not even 20, maybe 15 or 10 years, I think this will be part of our medical record in terms of getting your DNA sequence. I think the difficult part is analyzing what that means. For example, and I'm happy to say this, when I got my DNA sequence, I was actually looking for major mutations, just make sure I don't have major mutations. I have the aldehyde dehydrogenase variant that I knew without the DNA sequencing, right? Every time when I drank. But I also have a whole bunch of variants, and we weren't sure what those variants mean. And this is what we're stuck with right now. You get your DNA sequence, it comes back as a benign variant, and what do you do? Or potentially pathogenic variant, or even variant of no significance. What do you do with that? What I didn't show is we actually had a case in which it was referred to us, and I'll just briefly describe the scenario. So this is a VIP patient who came to us on the concierge clinic. I'm sure you guys have concierge clinic. We have concierge clinic. He got his DNA sequence, and it shows a potentially pathogenic variant in myosin light chain 3 that causes hypertrophic cardiomyopathy, right? But then when we did the echo on him, there's no hypertrophic cardiomyopathy. The heart looks fine, but he's not worried because he says it's potentially pathogenic. And so what do you do with that patient? Well, you could leave him alone, and he's a business guy, and let's say he flies from San Francisco to Frankfurt, and long flight, develop a DVT, deep venous thrombosis, and get a pulmonary embolism, and dies from it. And the doctor just says, well, they didn't pick up the PE. Young guy, probably just coded as a sudden cardiac death, right? The wife is actually a lawyer. So what's gonna happen? You're gonna get sued with that. And so alternatively, if you do all these testing on him, The patient gets scared and say, "Hey, doc, what's wrong with me?" And again, he's a business guy. And 2 o'clock in the morning, if he gets a palpitation, he's going to call his concierge doc and say, "Hey, doc, I have palpitation." And you're going to get a lot of phone calls. So this is the dilemma that we're dealing with on whole genome sequencing on everybody. This case was actually referred to our lab. We generated the iPSL. We differentiated to the cardiomyocytes. We showed that his cardiomyocytes look completely normal. We challenge it with a whole bunch of drugs, looks fine. In the end, we tell the patient this is a false call. And based on that paper, Clenvir, which is the most popular gene annotation, just included the paper and say this is a potentially benign variant as well. But this is a problem with the DNA sequencing right now. But I do think that in the future, everybody will have the DNA sequence. Yeah.
How does the iPSL approach to predicting drug response deal with the issue of variability in cellular response or tissue response based on physiologic differences or endocrine and exocrine effects of other organs and tissues? It seems like these are tested in isolation as particular tissues. And so how are these complex effects accounted for to predict the response?
Yeah, complex cell-cell interactions.
Yeah,
which can't be
yeah.
modeled
So
in cells.
I think 10 years ago, it's not possible to study the cell-cell interaction, but right now, with single-cell ATAC-seq, single-cell RNA-seq, you can then create organoids or engineer hard tissues, treat some type of drugs, and then do single-cell multiomics to see what is the effect of that drug on cardiomyocytes, endothelial cells, fibroblasts, and so forth. It's actually a perfect system right now to study cell-cell interaction, combining single-cell and combining 3D organoids and 3D tissues. I think the other question that you may be asking is, how does the iPSL recapitulate what happens to the patient? You can't. Right now, it's only for genetics, meaning that if I have an identical twin, and I'm healthy, but my identical twin is very, leads a bad lifestyle, obese, and multiple heart attacks, myocardial infarction, hypertension, diabetes, the iPSL will look the same among the two of us. It wouldn't have predicted that my identical twin has such a bad social lifestyle. Now, but it's actually a good model because of that, because for example, I'll give you one analogy that the trainees or the postdocs or faculty always do. Let's say, for example, you're interested in diabetes, and you get heart tissues from patients who have diabetes, and you will say, "Oh, these are the gene expression changes due to the diabetes." Let's say your neighbor is interested in hypertension, and your neighbor gets heart tissues and say, these are the gene expression changes due to hypertension. Another colleague is interested in, let's say, in hypercholesterolemia, and say this is the effect of high lipids on vascular endothelial cells and cholesterol. The reality is that the heart tissues that you get from our patients that we do a lot of multi-omic sequencing on, they have multiple hits already. They have hits from hypertension, diabetes, obesity, cocaine, a whole bunch of alcohol stuff. You can't tease out what's what. And for you to say that the expression changes I saw is purely due to diabetes is not true. So what you can do is take that patient, generate the iPS cells, differentiate it to the organoids, treat it with a diabetic condition, and they'll treat it with hypertensive condition, or treat it with some type of drug condition and try to match. And that allows you to tease out what signatures are specific to the insult that you are trying to study. Yeah. So you just have to understand its pros and cons. Yeah.
Time for one or two more questions.
So my question with regards to what you're modeling is really based off of gene polymorphisms. And I think in your last answer, you started to allude to this, but I think one of the challenges in the clinical trial in a dish essentially is how are you accounting for the epigenetics, the environmental exposures that we're all coming in touch with.
Yeah.
And I'm a mouse doctor, so I love having my controlled environments. And so I find people difficult because they don't, you know, they do a lot of different things and have a lot of different exposures that are going to epigenetically change those genetic profiles.
So I think, as I mentioned, with the analogy of the identical twins, the iPS cell would not have been able to predict that my twin brother has hypertension, diabetes, you know, obesity, because my twin brother decided to be on a hamburger diet 365 days a year. You will not be able to predict that, but you can, as I mentioned to you, you can try to tease it out by exposing it to different conditions to tease it out. Now, the model is, I think you have to know the limitation and also what it's able to do. A lot of times when we give it a drug, let's say, for example, I give a drug, half of you in this room have the drug, the other half do not have the drug. Within this half right here, the drug may actually work, but there's so much heterogeneity among the population that you're not selecting the patient who's going to respond. And if I just give the drug to you, and some of you may not have the particular receptor, particular enzyme that you're going after, it basically drowns this positive signal. And so as a drug company, and we do design all our drugs, let's say, for example, you have 10 drugs, and 10 drugs, they all tested fine in your mouse model, they all tested fine in your HeLa cells, and you go to drug number 1, fail. Drug number 2 failed. By the time you go to drug 3, the company will basically shut down the program. What we're proposing is that you could take all 10 drugs, test it on your panel, say, you know, 100 iPS cell lines or 1,000. If the drug works in 100 out of 100, it's impossible. There's no drug that worked that way. The scientists probably made up the data. If a drug works in 1 out of 100, we're not interested in it. We're looking for a drug that works in, say, you know, 30, 20 out of 100. Because if the drug block gene X, protein X expression, there's going to be a lot of heterogeneity among our gene X and protein X. And that's why we're using this model for it. It's also essentially give a sneak preview of how the drug responds by giving it to all of you guys that are here, but not you, but on your cells on the dish. Yeah.
Dr. Chopra, I'm going to give you the opportunity to ask the last question.
Joe, thanks for a wonderful talk. My question was really based on where you just ended, which is you talked a lot about repurposing of existing drugs, like your patient with a lamin C deficiency. What excites you about novel drugs? What's coming out? What can we look forward to from the work that you're currently doing and you can share in terms of novel drug therapy that may be on the horizon?
Vineet asked about, you used repurposed drugs, but what novel drug discoveries can you articulate?
Yeah, so I use, well, there's a couple of ways to think about this. One is that you could use repurposed drug that allows you to go to clinical very fast, right? Essentially getting IRB in the register on clinicaltrial.gov. A lot of times the repurposed drug doesn't have the biological activities that you want, so we will modify that drug to get a new compound entity pattern behind it. But then in that case, you would need to test the small animal, large animal, so that it's safe, start with the phase 1 trial, go back. But the FDA will give you faster clearance because they've seen the drug and it's almost like a pro drug. The third example, which is what you're asking, we have projects in which we actually do virtual library screen. As I mentioned, there's a slide. You give me a protein structure. My PhD was in pharmacology. 20 years ago, you look at it, you're doing a lot of pipetting. 20 years later, the protein structure, you know the polarity, you know the different groups, and then the computer can kind of say, this is an ideal drug that can fit into this pocket. You take this drug in its different software that could predict whether it crosses the blood-brain barrier, what is the half-life. It's all estimated. You then synthesize 20 or 30 of these drugs. And once you synthesize it, you have to go through the process of testing and make sure it doesn't kill the animal and so forth. That whole process with the new drug takes typically on average, 10 to 12 years. The literature will tell you that. But hopefully with these models, we're going to shrink the time period. Yeah.
Thanks very much.
Oh, thank you. Thank you. Yeah.